the West Nile virus, Usutu virus, and mosquito-borne Flavivirus. So Flaviviridae has the Flavivirus genus. They are single-strand RNA and they're enveloped, which means they have weak resistance. They need a tick or mosquito vectors to spread the virus, so they're also arboviruses. They are zoonotic agents and they are Eurixin viruses, which means they have a wide host range. The geographical distribution is characteristic to each disease, so certain viruses have certain areas. So for the West Nile fever, the occurrence, it's sporadic in the EU, Australia, Africa, and Asia. In the EU, it's sporadic in different lineages. In America, it's widespread in the country, so it's essentially worldwide. For the etiology, the West Nile virus is a part of the Flavivirus genus, which is part of Flaviviridae. It is part of the Japanese encephalitis serocomplex. They have a shared common antigen, and they have serological cross-reactions occur in lab diagnostics with the Japanese encephalitis virus, the St. Louis encephalitis virus, the Murray Valley encephalitis virus, and the Usutu virus. There are 9 genetic lineages, but lineage 1 and lineage 2 are pathogenic. Lineage 1 is found worldwide, and lineage 2 is found in Europe and Africa. For the epidemiology, it's a Eurixin virus. The natural host are birds. They maintain the infection. They have a high amount of the virus and they have a long viremia. Migrating birds are less susceptible. They have a subclinical infection and they play a role in spreading the virus over a large geographical location. Birds of prey are susceptible and they show clinical signs. Birds are the natural host and the dead-end hosts are horses, humans, it's a zoonotic disease, small mammals and reptiles. In these animals, there's a low amount of the virus and the viremia is short. Other susceptible animals are ruminants such as cattle, sheep, goats, and it also infects swine. Culex pipiens is a mosquito that spreads the virus to 60 different species. The disease is an arbovirus. It's spread by mosquitoes, so biological vectors, and transovarial transmission occurs. It's a seasonal disease and it happens in the summer when it's rainy and it's humid. For the pathogenesis, a mosquito inoculates the virus into the blood, and then the primary replication happens in the lymphatic tissue and the blood vessel endothelium, and then a viremia occurs, and the virus spreads to the visceral organs and the central nervous system. The virus infiltrates the central nervous system and causes degeneration, apoptosis, and inflammation. So for the clinical signs, in horses, 80 to 90% of infections are subclinical. The central nervous system signs are less than 1% of cases, but those signs are circling, incoordination, ataxia, and hind limb paralysis. The animals also experience depression, fever, and anorexia in animals with immunosuppression. There's a 90% lethality in animals that show clinical signs. Very few animals show clinical signs, but those who do, die. In geese, it affects animals between the ages of 3 and 12 weeks old. They have abnormal head positions, ataxia, seizures, tremors, and opitotinus. In dogs and cats, it's asymptomatic, and in sheep, they experience teeth grinding and ataxia. Wild birds are reservoirs for the disease, and wild bird mortality is observed before human and horse cases. In the pathology, we can see blood vessel damage, so petechial hemorrhages and fluid accumulation. We can also see lymphocytic infiltration and neuron necrosis and apoptosis, along with glia cell proliferation. For the diagnosis, look at the epidemiology, clinical signs, and post-mortem lesions. It's a notifiable disease, so if there's any clinical signs, lab diagnostics must be done. To detect the virus directly, do real-time PCR and IF. To detect the virus indirectly, do ELISA and viral neutralization. Viral neutralization is important due to serological cross-reactions. You also need to do a serosurvey, so distinguish whether it's caused by a vaccine or an infection. IgE, which is detected with an ELISA test, is found years after infection, so a vaccine may not be necessary. If IgE is negative, the horse needs to be vaccinated. If IgM is detected, this means it's a fresh infection. This means authorities must be notified. To prevent the disease, try and have mosquito control using nets and repellents, etc. There's also vaccination. There's an inactivated vaccine. It's given yearly before the mosquito season. It's given yearly because there's not strong immunity because it's inactivated. There is also a recombinated canary pox and West Nile virus vaccine. You can also try and stamp out infected birds. For the public health significance, there is seroconversion without clinical signs in 80% of cases. In immunocompromised patients, they experience fever, rash, hemorrhages, encephalitis, and there is no vaccine in humans. The Usutu virus is considered the little brother of the West Nile virus. It's very similar. The occurrence is in Africa and Central Europe. For the epidemiology, migratory birds are more susceptible. They get encephalitis, and encephalitis is also caused in humans. It's a zoonotic disease. Other mosquito-borne flaviviral diseases are Japanese encephalitis. It's zoonotic and notifiable. It affects horses, humans, and it causes abortion in swine. It's present in Japan and South Korea. There's also St. Louis encephalitis. It's in America and it affects horses and humans. It causes encephalitis. Then there's the Murray Valley encephalitis virus. It's in Australia and it's a zoonotic disease. It mostly affects children. Wild birds are the reservoir. Murray Valley encephalitis is a zoonotic disease. The next disease is the Wessels-Brons virus, which is also zoonotic. It's a phytopathogenic virus that affects sheep in sub-Saharan Africa. Another disease is Turkey meningoencephalitis. It causes economic and financial loss by decreased egg production and central nervous system signs. The next disease is Dug egg drop syndrome. It's found in China by the Baiyang-Diang virus. And the human mosquito flaviviral diseases are dengue virus, yellow fever, and Zika virus infection. With dengue virus, there's an antibody-dependent enhancement. The first infection is mild, with non-specific antibodies, and then infection again with old non-specific antibodies causes lethal shock syndrome. Yellow fever affects humans and monkeys. It causes liver dystrophy, which causes icterus. Dengue fever and yellow fever are found in the tropics. And for the Zika virus infection, in humans it causes rash and conjunctivitis. It also causes phytopathy. Both yellow fever and the Zika virus are spread by the mosquito Aedes aegypti.